Good day, viewers. This is 300 Plus Academy, where all we do is exams tutoring, ensuring that you ace your next and every exams. Okay, still on the business of NECO 2024 Biology Practical. We've looked at Group 1, we've looked at Group 2, we've looked at Group 3. And right about now, it's time to look at the last and final group, Group 4, talking about specimens K to specimen O. What we have in this group talks generally about 4 fruits and a flamboyant flower. So, what are the relationships we see? What are the drawings to take place? I personally think we may be drawing the transverse section of tomato fruit. I suspect, let's see, I'm suspecting drawing this. Then somehow, I'm also suspecting this. That's my, at least in the meantime, those are the things I suspected. I also suspected a ripe flamboyant flower here, but because it's the last specimen, and rarely do we have drawings on the last specimen, I think this is just going to be more of a table, and then we have to fill the table talking about the various parts of the flowers. Those are the relationships or the directions I am seeing things. But let's look at what is going to happen. So, as you can see on your screen, you have a ripe tomato fruit, quite expensive now. You have a ripe palm fruit. You have ripe traders fruit, a ripe mango fruit, and of course, a ripe flamboyant flower, or what you call Delonis regia. So, what are the possible questions after putting one or two things together? So, what are the possible questions we can have? As you can see on your screen, the first question says, classify specimens K, L, M, N. We should classify them. Anyways, they are all fruits. But when we look at specimen K is a class of fruit called a berry. Specimen L is a class of fruit called drupe. Specimen M is a class of fruit called cypsela. And specimen N, talking about mango fruit, belongs to the class of fruit called drupe, berry, drupe, cypsela, and then a drupe. On question 2, as you can see on your screen, we were told to state one agent of dispersal for each of K, L, M, and N, and also give reason for the dispersal method mentioned. Look at it again. State one agent that helps to disperse each of specimens K, L, M, and N, and give a reason for the method of dispersal that you have mentioned. For specimen K, the agent of dispersal is man or animal. So what is the reason why animal or a man will want to disperse this specimen K? Talking about tomato fruit, you can as well see that on the diagram on your screen. It's very simple. It is soft or succulent. It is, that's when I say succulent, it is juicy on the inside, and it is edible fruit. It can be eaten raw. Also, the fact that most times tomatoes are usually a red, brighter color, so it is attractive when ripe. The fact that it is attractive, it is juicy or succulent on the inside, and it is edible is the reason why man or animal is the agent of dispersal. When you talk about your palm fruit, what helps to disperse here? The agent of dispersal here can be a man, and it can be water. When you talk about a man, the fact that, you know, we want to get to the seed, to the inside of the palm fruit, that's the edible part, which is the seed. It's just like when you have your, let's say, your coconut, you want to eat the whitish seed on the inside. That's the edible part. So, and that is very sweet, come to think of it. So the sweet edible seed is why a man will want to disperse it, is why we will want to pluck this and in the bid to do that, to begin to disperse that. Also, I mentioned water here. The reason why is this: the mesocarp. If you look at the diagram there, you must have seen the mesocarp. That is the airy or the fibrous part we have to remove before we get to the stony endocarp. That's what we call the shell of the coconut. In this case, we are looking at the palm fruit. So the thing is this: the fibrous mesocarp giving it buoyancy. If you place a whole palm fruit on a water surface, or let's say something like coconut, it is going to float because of the fibrous mesocarp giving it buoyancy. So water can as well stand there as agent of dispersal because of the fibrous mesocarp that gives it buoyancy. It allows it to float on water. The next one, traders fruit. Take note of this traders fruit. Somehow I suspect you might be drawing the traders fruit. Take note of the airy purpose. Take note of the airy pericarp with the seed as well. Very important. Okay, so talking about the traders fruit, the agent of dispersal here is the wind, and it is very simple, simply because it is light and there is this airy purpose that acts as a parachute, so that it's very easy for wind to carry that fruit. So that is the agent, and that is the reason why we are saying wind is the agent of dispersal. Here talking about mangrove fruit, it is man or animal, just like specimen K. Why is it that it is man? It is because it is succulent, juicy mesocarp, and then the fruit is edible. That's why a man will want to do that, and other animals too can do same. And also, it is attractive. It is attractive when you look at your mango, green, yellow. It's attractive. That is why a man or animal will be the agent of dispersal. And that ends question 2. Talking about question 3, we're told to state 5 structural differences between specimen K and specimen L. Structural differences here means the differences we can observe either by looking at it from the outside, using a scalpel, cutting through, and looking at what is on the inside. As you can see on your screen, talking about your tomato fruit specimen K, it is many seeded. It has many seeds, but for your palm fruits on the inside, it is having only one seed. Now the epicarp here, or the epicarp here is succulent. It's juicy, but in the palm fruits, the epicarp is dry. The epicarp is dry. By epicarp here, you can see in the diagram on your screen, the epicarp for a palm fruit is not even edible. It is dry and not edible. But for a tomato fruit, that's talking about specimen K, it is soft and edible. It is succulent. Now the mesocarp for tomato is succulent and soft. Talking about the mesocarp, you can see just on the inside, it is very edible and succulent. But for the palm fruit specimen L, the mesocarp is fibrous. Also, when you talk about the tomato fruit, if you bring out the seed does not contain any liquid, juice, or any water. If you bring out just one seed, it doesn't contain any liquid. But the seeds, talking about the palm fruit, when you grind it, or let's even say like a coconut, for example, it contains liquid or juice on the inside. In fact, what we try to extract from the seeds of most drupe is the juice that they contain, the fluid or the liquid that they contain. When you look at the tomato, it is the tomato fruit. It is usually red. Sometimes it can be yellow and it can be green depending on which one you're given on the day of the exam. But when you look at your palm fruit, it is somewhat brown. Also, talking about the okay, I've talked about the mesocarp. The mesocarp for the tomato fruit is succulent and edible. The mesocarp for the palm fruit is not succulent. The tomato fruit, the seeds are small. It is small seed. The palm fruit is a big seed. Now, when you look at the tomato fruit, you see the endocarp. You have to look at the endocarp on the inside. The endocarp and the mesocarp. The mesocarp and the endocarp are fused. But when you look at your palm fruit, the mesocarp, look at the diagram very well. The mesocarp and the endocarp is not fused. That's why we can just separate them from one another. You remove the fibrous mesocarp, you're going to see the endocarp. They are not fused together. In fact, they are totally different. But in the case of tomato, you cannot separate the endocarp and the mesocarp. They are fused. That's just about the difference between specimen K and specimen L. What about if it had been for us to differentiate between specimen L? What if the differences was to for us to differentiate between specimen L and N? This is just thinking in a broad way. What if it had been specimen L and specimen N? What are the differences we should be looking out for? The first one, you're going to see that on the palm fruit, there's going to be a scar left by the style. You can see that scar left by the style, but for the mango fruit, that specimen N, you're going to have a fruit stalk left. You can see that in the diagram. For palm fruit, you have a fibrous mesocarp. For mango fruit, you have a fleshy mesocarp. That's the part that we eat there, the edible part. Also, the endocarp of the palm fruit is stony. That's even the part we need to break open before we get to the seed. The endocarp is stony. But for the mango fruit, when you look at the endocarp, that's before the seed on the inside, it is just woody endocarp. Number 4, the mesocarp here being fibrous is not is non-edible. The mesocarp and the epicarp. Look at the diagram. The mesocarp and the epicarp, that's on the outside, the outermost epicarp, and then the mesocarp just immediately after the epicarp. They are non-edible. But when you look at the mango, the skin, that's the epicarp, and then just the edible inside, that's the mesocarp. They are all edible. And then number 5, the seed for the palm fruit is edible, whereas the seed for a mango is non-edible. So those are just about the differences between specimen L and specimen N. Like I always say, a biology practical is not complete without drawing. So what are the possible drawings that we can have here? Looking at question 4, I'm going to be displaying the drawing on your screen so that you just can look into that. Question 4, we were told to make a drawing 10 centimeter to 12 centimeter of the transverse section of specimen K. Transverse section means you're going to get the tomatoes, you're going to cut horizontally. You're going to get the scalpel, you cut horizontally. That's how, then you open it up. That's how you get the transverse section of specimen K. That's of a tomato fruit. if the question says longitudinal section, then you're going to cut longitudinally this way. What you need to draw and what you need to label is displaying on the screen with the labelings right about now. You're looking at the let's look at the transverse section. You're looking at the soft epicap on the outside. You're looking at the placenta on the inside. You're looking at the fleshy mesocarp just after the epicap. You're looking at the juicy endocarp, and you're looking at the seeds. That is just about all. If it is the longitudinal section, you can see the stalk. You can see the stalk with the calyx there. You can see the soft epicap. After the epicap is the fleshy mesocarp, then the juicy endocarp, then the seed, the remainder of the style, and then the placenta, you know, at the center, talking about the fact that it has what we call axial placentation. We're going to talk about the placentation later, but what you need to draw is exactly what you have on your screen. What else can we be told to draw? What else do I suspect? I think I suspect this, and then I'm suspecting strongly your treatise fruits. Yes, I suspect drawing the transverse or the longitudinal section of a ripe tomato fruit, and I suspect drawing a ripe treatise fruit with its airy purpose. Let's look at that. As you can see on your screen, you can see the drawing for specimen M. Talking about treatise fruit, you can see the scar left, the scar. You can see the fruit itself, and on the you can see the fruit, and then you can see the air, and then you can see the purpose. That's the airy purpose. Just about 4 labelings, and you're done with things. You know, look at the diagram very well. Look at the fruit itself, very light fruit, and look at the parachute like airy purpose. So I think I suspect that as well, and that just about ends that. Recall your biology practical is incomplete without your drawing, so take note of that. I think the best thing will be for you to pause this lecture video and get your pencil, draw, measure it 10 to 12 centimeter, or if it's 8 to 10 centimeter, do the measurements and sketch, and that way you're ready for your exam. So that ends that. So moving on to question 5, we were told to state the type of placentation that is present in specimen K, L, and N, and we should name one example of fruit with similar placentation. Talking about specimen K, the placentation is axial, and examples of fruits or plants that exhibit similar placentation, you're looking at orange, garden egg, pepper. When you talk about specimen L, that's the palm fruit, it is a free central placentation, and other examples of fruits that exhibit the same placentation, you're looking at water leaf, you're looking at coconut. Specimen N, that's a mango fruit, it also exhibits axial placentation. So you can just pick from any of the example here and slot it in here correctly, and that ends question 5. So on the next question, talking about question 6, we were told to carefully examine specimen O, and we were told to copy and complete the table using our observation of specimen O. Normally, specimen O is a flamboyant flower, or what we call flame of the forest. You can see in the diagram on your screen. Normally, you're going to have your petals. You're going to have your sepals. You can see the sepals just after the receptacle. You're going to have your petals, and then you're going to have your filaments. Yes, talking about the filaments that holds the stamens. Yes, the that holds the anther, the filament that holds the anther, and the anther and the filament together constitute the stamen. You can see that in the diagram, and then you're going to see your stigma. How many stigma are we looking at here? The stigma, as you can see, is usually one in that diagram. The stigma is usually one. And then the filaments, when you count the filaments holding the anther, it's usually on an average of about 10. And then talking about the petals are usually 5. So you count that the bright colored petals. And then when you look at the sepals, just after the receptacle, it's also usually 5. You can see that in the diagram. So you use that to complete the table. You can see the table, what your answer should look like. Talking about the sepals first. The sepals is just immediately after the receptacle. You can see it is green on the underside, and then it is red or orange on the upper side. You can see that. Then talking about the petals, it depends on what you're supplied with. If it is red, put it there that is red. If it is cream, put it there, because when we talk about flame of the forest or flamboyant flower, the color varies. It's not that it must be red at all times. So you put the color, you can see that they look at the position of the petals. Talking about the filaments, you have to also, the filament is exactly what is carrying the pollen grain that's in the anther. So you have to put the color there. If it's red, put red. If it's pink, put pink. If it's milk, put milk color. And then the stigma, you put the color as well, the one at the center, just immediately after the oval, after the ovary. You're going to see the stigma. You're going to see the stigma, a long style to the stigma. Put the color there, and I think that's what I suspect for specimen O, a case where you're given the table and told to list the number of the floral parts and the color of the floral parts. That ends that. So what else can they ask us on this particular specimen? Can we be told to draw? Yes, we can be told to draw. Imagine when they tell us to cut into half. We should cut the flower into half and draw. So what are we going to be drawing? So look at what you have here. When they tell you to cut into half, it now means that the sepal is going to reduce from 5 to about 2 or 3, and the petal is going to reduce from 5 to about 2 or 3. Even the filament is also going to reduce. So if you are told to cut into half, you cut into half and you draw what you can see. What if you're told to do that? What you should be drawing is exactly what is displaying on your screen as of now. Also, imagine talking about the drawing. You can be told to carefully detach the petals, except the special. You can see that, except the special petal. Make a large label drawing 10 to 12 centimeter long of the remaining floral parts. You can see that. Look at it. They say carefully detach the other petals except the special petal. And then we were told here to make a large label drawing 10 to 12 centimeter long. When we say the special petal here, the special petal is just the one immediately after the sepal. If you look at that diagram displaying now on the screen, that is what we call the odd or standard petal, and that's what is called the special petal. So in this question, we're told, you can't be told to detach the other petals except the special petal and make a large label drawing 10 to 12 centimeter long of the remaining floral part. So what should your answer look like is exactly what you are seeing on your screen. You can see the anther, you can see the stigma, you can see the filament, you can see the style, you can see the special petal itself, you can see the sepal, you can see the ovary, and you can see the receptacle. And that's just that about that. That's what you can have on specimen Do we have any other question? No. And with that, we have come to the end of this series on Biology 2024. We've looked at Group 1, Group 2, Group 3, and we've just concluded on Group 4. Make sure you practice all of your drawing, and I tell you, you cannot go wrong other than ace that exam. From myself and the entire team, it's bye for now.